This is Stanford Engineering's The Future of Everything, and I'm Russ Altman. You want to help shape future episodes of the podcast? Leave a review and tell us what you would like to hear next. We'll see what we can do. In fact, today's episode with Suzanne Pfeffer was suggested by a reviewer who said, how about a little bit more on Parkinson's disease? It affects millions of people. Many of us know someone, a loved one or a friend who suffers from the disease. So we found a local expert, Dr. Suzanne Pfeffer, and we're going to be talking about Parkinson's disease today. In addition, today we're continuing our feature, The Future in a Minute. At the end of my conversation with Suzanne, I'll ask her some rapid-fire questions, and she'll give us some rapid-fire answers. And again, before we get started, remember to rate and review the show. It helps us, and it helps others learn about the show. Today, Suzanne Pfeffer from Stanford University will tell us that our understanding of Parkinson's disease at the molecular level is increasing, and we have a better understanding of what's going on in the brain. So much so that we can now detect early signs of Parkinson's 20 years before the big bad symptoms really occur. This is leading to new opportunities for treatments that are quite exciting. It's the future of Parkinson's disease. Parkinson's disease is a terrible disease that affects millions of Americans. It's thought of as a motor disease, a motion disease. People get problems with walking smoothly. They get a tremor. They get a lot of motor disturbances, and it can progress over the years to become worse and worse. The treatments are not very good. There's deep brain stimulation, which can last for about 7 years, and there's medications which last for about 5 years, but then the side effects of those medications outweighs their benefit, and we often have to stop them. Well, Suzanne Pfeffer is a biochemist and a professor of biochemistry at Stanford University, and she's an expert at the molecules that go wrong in Parkinson's disease. She's studying them, and she's learning more about what goes wrong in the disease, and importantly, she's generating hypotheses about new ways to treat the disease, not just when the symptoms come on in your older age, but very early when you're first showing signs that you might have a risk for the disease. It's very exciting, and she'll be telling us about it. Suzanne, you're a biochemist. What led you to make the decision to do deep dive in a disease like Parkinson's disease?
I was actually really lucky. So we were studying a small family of proteins called Rab proteins, and they control how proteins are put in the right place in cells. And our collaborator, Dario Alessi at the University of Dundee, made a discovery that an enzyme that's really important in Parkinson's makes a change to my favorite protein. And he didn't know about my favorite protein. And out of the blue, I got a phone call. Suzanne, we need your help. We want to understand how your proteins are part of Parkinson's. And that was a totally life-changing experience. And he's the most wonderful collaborator. So now we're celebrating our 10th year of direct collaboration. And I have learned so much. And the work is really so meaningful. So it was a serendipity that his discovery led to my set of proteins, and here we are.
So biochemists, just to stick on this topic for a couple minutes, biochemists are known for their kind of commitment to basic science. You know, in fact, you were probably studying these proteins and they were probably interesting to you biochemically and they did interesting things. Is it a big, hard decision to say, I'm now going to think about this as the understanding a disease and maybe even getting involved in the development of treatments, where before it was just the pure kind of curiosity-driven research? I'm just very interested in your decision about how to balance those two. I'm sure they're both passions for you.
So the reason I'm a biochemist is I want to understand how proteins, these tiny little molecules, make us able to do all the things that humans can do. So fundamentally, that's the reason I'm interested in these molecules. But so any connection to disease makes the work all the more meaningful, because it's not just working out just the picture of a cell and what it is. It has real consequences for people. And that has been one of the most wonderful parts of the last 10 years, is the work is super meaningful.
Great. Okay, so I think it would be good for us to have a little tutorial on like what we need to know about Parkinson's disease to appreciate the current frontier challenges and the work that you're doing. So maybe tell me a little bit about the disease. How should we think of it? What do we know about the causes, just for starters?
Okay, so let's start with the causes. So most of the causes are probably environmental. We absolutely know that pesticide exposure can greatly increase your risk of Parkinson's, even trigger it directly. And so that's bad news for farmers and people that live in regions. Even living near golf courses may be dangerous, right? We never think of that. You think of getting a retirement home, a condo facing the golf course, well, that may not be the best plan.
So these are actively used pesticides, not just
Yes,
historical
and in
ones.
the U.S., they have still not been outlawed. The Europeans are ahead of us, but it's our environment is for sure linked to Parkinson's disease. Now, for scientists, that makes it difficult because a person comes at age 65 with disease. We don't know where they've lived and what they might have been exposed to. So when there are genetic causes of disease, it gives us an inroads to study the process. And especially if those genetic forms, the kind that are inherited in your family from your parents, your grandparents, when those forms come on in a way that doctors identify very similar to the ones caused by pesticides, it gives scientists a really important clue that we can zoom in exactly on that specific form of disease, understand what's gone wrong with that particular gene change to cause the disease, and then see how does that link to the other forms of disease.
So is it the case that some people have the genetic predisposition to Parkinson's, but that other people just have the environmental exposures and perhaps don't have the genetics and still get the disease?
Absolutely. But what's important is even if you have a genetic predisposition, it's not a guarantee that you'll get disease. So we call this penetrance. And so in some populations, in some mutations, it may be you have a 50-50 chance of getting Parkinson's. So then if you have that 50-50 chance plus exposure to pesticides or smog or other unknown environmental factors, also the chemicals used for dry cleaning, those can be problematic so workers who've worked in a dry cleaning store, for example, may be of increased risk. Also, if you're a boxer, you got hit in the head or too many football concussions, that can also increase your risk. So it's going to be a combination.
Okay, and so tell me a little bit about what actually goes wrong. We know it's a brain disease. What happens in the brain?
So, and this is really important, and it actually guides our work. So the two most common forms of neurodegeneration in people are, number one, of course, Alzheimer's disease is the most prevalent, but the second most common one is Parkinson's. And we always try to say, what's the difference between them? So Parkinson's is classified as a movement disorder. So you may know someone who has what we call a resting tremor. That's what people usually think of, where a hand at rest is showing a tremor only at rest, or it may be the head has a shake, or the foot has a shake, or the tongue. So people think of Parkinson's as being a tremor disease, but it's actually a movement disorder. And what it is, it's related to how smoothly you're able to initiate movement, stop, keep your balance, and not walk rigidly. So walking slow and highly rigid is a very predominant sign of actual Parkinson's disease beyond the resting tremor.
And have we isolated the areas of the brain that are preferentially damaged?
Yes. So again, this is what we're trying to understand. There's a specific circuit in the brain that controls movement, this starting and stopping of movement and the rigidity. And this is called the nigrostriatal circuit. And in Parkinson's, it's the nigrostriatal circuit that is vulnerable, that is the cells die and are unable to do this process of controlling movement. And that's different than Alzheimer's. Alzheimer's can be different regions of the brain, and people with Alzheimer's present in very different ways. So some people may get very angry, very hostile. Some people just lose their memory. Some people have trouble swallowing. So again, in Alzheimer's, it could be different regions of the brain. And we're trying to understand in Parkinson's, why is it specifically this circuit that's especially vulnerable? And that's the focus of our work.
Great. And one more question, and this comes from my personal, you mentioned that many people have family and friends, and in fact I did,
Yeah.
my uncle. Two of the diseases that were bandied about when they were making a diagnosis on my uncle was both Parkinson's disease and Lewy body dementia. So can you just tell us a little bit about what are the differences and similarities? Are these the same disease or are they different diseases? It was very confusing to the family since both of them were bandied about.
So Lewy bodies are these accumulations of a protein called synuclein that are seen and typically found in Parkinson's disease. Now, late-stage Parkinson's often will develop into Lewy body dementia. And often there's a crossover in symptoms where you can have Parkinsonian symptoms in outright Alzheimer's. But let's just focus to keep it simple.
Yeah.
In Parkinson's, the brains you see have Lewy bodies that are full of synuclein, and it's not at all for sure if they're the cause or the consequence, but they are characteristic of most, but not all forms of Parkinson's, and people would like to call it a Lewy body disease. But Lewy body dementia is where you have Lewy bodies in other regions of the brain that are consistent with the dementia, the inability to remember things and to have that kind of brain function.
Great. Well, thank you very much for this tutorial. So now we all have a kind of a shared understanding of these circuits. But you made some intriguing comments about some molecules, and I think we should try to understand where you are in your research and what we've learned. So tell me a little bit more. You said there were some molecules that are involved in, I reviewed your papers, what they call the trafficking of proteins, where it tells the proteins in the cell where they need to be to do their job. So tell me about how those things are involved with the disease, and then some of the other molecules that are important.
So we were talking before about how scientists use genetic forms of disease to really understand what's going on. So we studied two proteins that are, when mutated, the most common risk factors for Parkinson's, and that's an enzyme called leucine-rich repeat kinase 2, or LRRK2. We call it LRRK2.
LRRK2, nice and easy to say.
Yeah. And the other is a lysosomal enzyme. I'll say what that is. It's a protein called GBA, glucocerebrosidase. And if you have, so the problem with the LRRK2 is you have a mutation, LRRK2 becomes what we call hyperactive. So LRRK2 makes a change on a protein. It puts a little charged molecule, a little charged piece, a little modification, changes the structure of a protein. And if the mutation does this too often, okay, too much of it.
Right.
And so when it happens too much, what happens is it blocks a process in the brain that we discovered, which is generating a little antenna on the surface of the cell. And that antenna is called a primary cilium. It's involved in signaling. So it's listening for signals, chemical signals, that require the antenna that make this cell change.
It's a very fun idea that cells would have antennas, and it's very attractive because, of course, they need to communicate. So it's fun that there's actually kind of like an antenna.
It's a physical antenna. It's pretty, and they're beautiful little structures. Anyway, they're on nerve cells and they're on the supporting cells around the neurons. And what we discovered specifically in the nigrostriatal circuit, the part that's important in Parkinson's, the cells talk to each other. So here, this is a cell that secretes dopamine. These are the ones that die in Parkinson's. What they do is they send out a long process, a little extension, we call it an axon, to another region of the brain, and they secrete a signal when they are stressed. And the antenna has to receive that signal, and that makes this cell produce protective factors that make this cell happy. So normally, if this guy's stressed, the job of this one is to send happiness factors, so these cells are happier. They're better able to deal with stress.
Right.
And what we discovered is if you have the hyperactive form, the 2-active form of that LRRK2, these cells lose their antennas. So this guy's stressed. He's sending a signal. This guy can't receive, doesn't know it's happening. It doesn't have an antenna, so it can't send back those protective factors. And so then this cell starts to retract its axon as part of its process to die. And that's something we want to stop in Parkinson's disease. So we're trying to understand how we can rescue that antenna so it can get the signal to send back the neuroprotective factors.
Now, you mentioned these two proteins, the LRRK2 and the one that begins with a G.
GBA.
GBA, thank you.
Yeah.
What are they on the part of are they part of the antenna, or are they part of the distress signal, or both?
Good question. So what we know is that LRRK2 blocks the process of the antenna being there. So in LRRK2 mutation, half of the cells lose their antennas. In the GBA, the cells keep their antennas, but they're not functional. This antenna's broken. It can't sense the signal. And we showed that the GBA mutation changes the structure, so it can't signal anymore. So it's there,
Gotcha.
but it can't do its job.
So, one of the things that you said, you gave a scenario of a 65-year-old, but I know that there's a question about when these pathological changes in the brain start, and what do we know about this, and are we seeing these malfunctioning cilia, if you will, primary cilia? Are we seeing them very early, or do we understand how quickly that happens in the disease process?
We don't know that yet in humans. So far, we've only looked at brains from people who passed away at the age of 85. And we clearly see in all forms of Parkinson's, not just the genetic forms, but the kind that we don't know the cause of that are likely due to environment plus some genetic factors. We see the antenna go away. They go away more in Parkinson's than in control brains from people of the same age. And so we don't know how early in humans. In mice, it's pretty early. In mice, it says, it's at 8 weeks, we can see the problem. But remember, only half of the cells lose their antenna. So the other half are still there. And what we think is happening, this is kind of like it's a slow process. So you have half the cells, maybe you're making half as much neuroprotective factors, but by the time you're 65, it isn't enough.
Yeah.
Because it's been all those years, a slow process. And this comes back to earlier symptoms of Parkinson's disease. So we know in Parkinson's, 20 years before you have a resting tremor or rigidity of walking, 20 years earlier, you have a couple of symptoms. One is constipation. Now, anybody can have constipation. That's not proof you're going to get Parkinson's. But you may have something which is called REM sleep behavior disorder. This is where, when you're sleeping, you're acting out your dreams. And these are often sometimes violent dreams. So you will be kicking and punching. And your partner will say, hey, you're waking me up. You're kind of dangerous over there. What's going on? If you have REM sleep behavior disorder, there is a very high probability, and this is 20 years before anything else.
Wow.
There's a very high probability that you will get Parkinson's. It's not it's something I wouldn't want to be diagnosed with.
Yes. So the constipation is not very specific. But the REM disorder is,
and also loss of smell. So just like in COVID, where many people lost their sense of smell, an early symptom of Parkinson's, 15 years before you have any of the movement symptoms, will be a loss of sense of smell. And most people don't really notice when they're losing their sense of smell. They may start wearing stronger perfume because they don't realize it, but it's very subtle unless it's really tested. And something for the future, I hope, is that everyone, when they go for their annual physical exam, will take a scratch and sniff test, which costs $1 or something to offer people. It's very inexpensive, but it's something we should be monitoring, whether people how sensitive and able they are to sense.
Yeah, the last few minutes of your comments, and I know you've written about this, make me understand why we shouldn't think of Parkinson's as something that just hits us when we're old, but there actually probably should be, as part of your primary care, a certain amount of surveillance for these different symptoms that you've just described.
Absolutely, yeah.
This is the future of everything, and we'll have more with Suzanne Pfeffer next. This is the future of everything, and I'm Russ Altman. I'm speaking with Suzanne Pfeffer from Stanford University. We've been discussing Parkinson's disease, but now we're going to focus on treatments. What are the current treatments, and what's coming down the pike that looks promising? Don't forget, at the end of the episode, I'll ask Suzanne my rapid-fire future-in-a-minute questions, and she'll give us her answers. So, Suzanne, what is the current state of treatment for Parkinson's disease, and what's good and what's bad about the current treatments?
So one of the challenges and limitations, the treatments right now are just treating symptoms. So basically, you would be given something that will help the resting tremor or help you be able to coordinate movement a little better. But the problem is that after about 5 years, with time, you need to increase your dose and increase your dose and increase your dose. And then at some point, it has a bad consequence, which is it caused something called dyskinesia, which are these movements which you can't control. And it's very embarrassing and very uncomfortable for people to have those. There's also great success with something called deep brain stimulation, where they actually place an electrode into the brain to try to counteract some of the signals. And that works very, very well for about 7 years. But then at some point that also stops. So we need something that's not temporary for these people, because you can live a long time with Parkinson's, and the symptoms, you really would like to be able to button your shirt when you get up in the morning, or be mobile and independent, and not worry about a freezing of gait where you basically cannot move forward. I mean, that would be terrible if you're trying to go about your daily life.
Yes.
So you really would like to do better. So we have to do better, not only to help with symptoms, but also to slow the progression. And that's really the limitation at the moment.
In our previous conversation, you were mentioning these symptoms that can come 20 years early: the sleep problem, the smelling problem, and I take it that if you did have all of these symptoms and you told your doctor, and your doctor was then worried about Parkinson's, none of those medications that you just described would really be very good 20 years early, would they?
That's correct. So, for example, they will prescribe melatonin to help you sleep better, but it's still not going to address the underlying cause of the symptoms. So it's a challenge. So we need to learn what's going on at the earliest parts of disease, and try to block it, and maybe even reverse it if we can.
Yes, and because I like to prepare for my interviews, I did look at many of your recent papers, and there are some very promising directions on some of the molecules you were discussing earlier. So can you tell us what's the status of these potential new treatments based on the molecular mechanisms that you and your colleagues are working out?
So I told you about LRRK2, and LRRK2 being important for a cellular antenna. And there are now 32 biotechs in the space of trying to identify an inhibitor that will bring it down to normal levels. I told you it's too active, but all of us have a certain amount of activity. So we just need to inhibit it or bring it down in its activity a little bit. And one phase 2b clinical trial just completed with the LRRK2 inhibitor. And there are several others that will be read out fairly soon. The first one, unfortunately, the good news is it showed very good safety profile, but it didn't seem to benefit the people that were included in the trial. And my own feeling is I think it was the wrong group of people. We know that there are people specifically where we know they have too much LRRK2, and that's the best patient population. They're hard to recruit for the trials, but the next trials coming out that we're going to be hearing about, we're very careful to only use those populations, and my fingers are crossed. I'm very optimistic that there will be benefit for those people. Also, I think a limitation of the previous trial is it could use more sensitive methods to monitor any success in the trial. And of course, it's a problem. These kinds of if you live for 20 years with Parkinson's, the changes year to year are not going to be as great as if you live a very short time. And so the readout we'll have to we want to get more specific so we can actually see the benefit of these compounds.
So for the phase 2 trial that showed good safety but wasn't able to show a lot of change, was that done on elderly advanced disease, or was it done on these folks who have the early symptoms? What kind of patients did they use?
They were patients that were relatively newly diagnosed, but they didn't necessarily carry the predisposition, the mutation in LRRK2. And so if it's of unknown cause, we can't be sure that this is what we call precision medicine. It's a precise drug that will help specifically people with this mutation, but we can't be sure it'll help everybody.
Yes. So for people who don't think about clinical trials every day, just to restate what you just said, the idea is that Parkinson's disease might have subtypes. There might be subtypes that are particularly well suited to a certain drug. And if you mix up the people who are very well suited to people who are not very well suited, it may overall look like your drug is a loser, but actually there's a fraction of that population who got a great benefit. And so that's why I presume that's why they were redoing these trials now with a more focused way. And in terms of the mechanism, the mechanism is actually, I think you've told us everything at least to a certain level. You were saying before that the LRRK2 mutations sometimes are making these changes to proteins and they're doing it too much. And so I guess these molecules, you might have even said this word, are inhibiting the LRRK2 so that it's not quite as robust in its enthusiasm for making changes to other proteins and kind of brings it down to a normal level.
That's absolutely. But we had a really exciting result last year, which is we took some of these drug candidates, and a particular one, and we fed it to mice that have too much LRRK2 activity, the super enthusiastic LRRK2. We fed the mice for 2 weeks and we looked at the brains and there was no change. But we fed them for 3 months. And what we found was the antennas grew back. The cells began again to make those neuroprotective factors. And this cell, which was retracting its axon on its pathway to death, actually came back.
Wow.
And we actually saw restoration of signaling, talking between these two neurons, restoration back to normal. So it wasn't just stopping disease, it was reversing the process. Now, the thing is, these mice are a model of early Parkinson's. And so our hope is that those same reversal of symptoms will help people if we can get the drug to them at the earliest stage.
Yeah, that's why I asked you about the population that was tested, because it would be hard if somebody is, 40 and just having initial symptoms. It might be hard to get them to be very excited about being in a clinical trial because the level of disease is not so great. They're not really aware of it yet. And yet that might be the group that would eventually benefit the most.
Absolutely.
Is that correct logic?
Absolutely. Absolutely.
Okay. Well, so this is great. So in the last 2 minutes, I just wanted to loop around because I know you're passionate about this. You're a biochemist and you were studying proteins that you found to be fascinating and important to understanding basic neuroscience and other fields. And then all of a sudden, you got a call from somebody who was a Parkinson's researcher, and now you described this 10-year journey. This is really a great story about the value of basic science, because when you started studying these RAB proteins that you mentioned earlier, I think you mentioned them, but I know they're RAB, you probably weren't thinking that this was going to be a Parkinson's disease relevant study. So what do you take from that whole experience, and what would you tell people who maybe don't understand why you're doing research on molecules that you don't even know what disease they might be involved with, or if they're even involved with a disease at all, right?
So I've lived both lives. I've lived a life where I study a specific disease for the past 10 years. And the 30 years before that, I studied a general process. And I could tell anybody it's important in cancer, in immunology, in neurodegeneration. We could make the story relevant to all these things, but it's a completely different world. And today, what I would say is I would rather pick a disease and study the molecules, because I can sit on an airplane and talk to the person next to me and say, I work on Parkinson's, and I can meet with people who have people with Parkinson's, and it's just so much more valuable and meaningful to me as a person. I'm making a difference. I am making a difference in people's lives. And sure, I was doing that before, but now I'm doing it in a very concrete way. And as a researcher, we work days and nights and hours and long. So it's a full dedication. I think the extra benefit of studying a disease is just so satisfying, personally satisfying.
Well, yes. And thank you for your work, which is really giving many of us who have family, friends who've suffered from this disease a lot of new hope. Before we finish up, I just wanted to ask if you're ready for our segment that we call Future in a Minute.
I'll try.
Okay. So as you know, I'll ask you these questions, and I'll try to ask them quickly, and then you give me your answer. Okay. What is one thing that gives you the most hope about the future?
I think our ability to integrate predisposition to disease, the earliest symptoms, and newly discovered disease biomarkers should really make it possible for us to address neurodegeneration and stop it before it develops. It's a very exciting time.
What's one thing you want people to walk away from this episode remembering?
I think people don't understand about how cures are found for disease. Fundamental research is critical for our ability to identify ways to help people and cure disease, for any disease, both for treating symptoms and for actually curing the disease.
Aside from money, what is the one thing you need to succeed in your research?
I'm going to tell you two things. First of all, you need a talented team. It takes a team of people to do the work, but we also need brains. And it's hard to ask people to donate their brains, but please consider it because it helps us be sure that what we study in the lab is directly related to what we see in human disease.
If all goes well, what does the future look like?
I think the future looks like much better treatments for Parkinson's, and hopefully we'll be able to stop disease progression in its tracks so that we have a world where people really don't have to suffer from Parkinson's disease.
And if you were starting over again and you needed to get your degree or your certification in a different discipline, what would that be?
Well, as you've said many times, I'm trained as a biochemist and a cell biologist, but I study whole brains. And so I think I could really benefit from training as a cellular and systems neuroscientist to really understand the consequences of molecular changes in the context of an entire organ that's as complicated as the brain.
Well, thank you very much, Suzanne, for your time on the future of everything.
Russ, thank you. It was a pleasure talking with you.
Thanks to Suzanne Pfeffer. That was the future of Parkinson's disease. Thank you for listening to this episode. Don't forget, we have a back catalog of more than 300 episodes. You can spend all day listening to the future of pretty much anything. If you like what you hear, follow the show. Press that button. You'll be alerted to new episodes and you'll never miss the future of anything. You can connect with me on many social media platforms, including LinkedIn, Threads, Mastodon, and Blue Sky, where I'm at RB Altman or at Russ B. Altman. You can also follow the School of Engineering at Stanford, at Stanford School of Engineering, or more briefly, at Stanford ENG.